Welcome to the latest Neuro Shorts podcast, brought to you by MedSynapse. Neuro Shorts is a weekly program covering a wide range of clinical and scientific medical content that offers enriching insights into the world of neurology. In today's podcast episode, we will share details on the results of the study titled Development of Novel Clinical Examination Scales for the Measurement of Disease Severity in Creutzfeldt Jakob Disease, which is published in the Journal of Neurology, Neurosurgery and Psychiatry on 12 January 2022. Creutzfeldt Jakob disease, a human prion disease, can be classified as sporadic, inherited or acquired, based on the etiology. An enormous heterogeneity in clinical phenotype, duration, and histopathology is observed within the groups. Well-designed clinical rating scales are required to interpret and manage the observed variability by providing quantitative measures of disease-specific change, as clinical trial eligibility and stratification criteria, outcome measures, or for prognostic modeling in Creutzfeldt Jakob disease patients. Rash analysis was used for construction of interval scales, which measured composite cognitive and motor dysfunction, from pooled bedside neurocognitive examinations, from October 2008 to December 2016, collected as part of the prospective National Prion Monitoring Cohort Study. 144 assessments from 106 patients were included in rash analysis to develop the motor scale. All included patients were diagnosed as having probable sporadic Creutzfeldt Jakob disease by consensus criteria. 231 assessments from 155 unique patients were included in the cognitive scale development. In addition to gait, which is reliant on multiple motor systems, the final motor scale included items reliant on pyramidal, extrapyramidal and cerebellar systems. Thus, the motor scale can be used to infer the relative pattern of progression in motor dysfunction for patients with sporadic Creutzfeldt Jakob disease. The final cognitive scale included measures of executive function, language, parietal lobe function, visual perception, recognition memory and attention memory. Individual cognitive and motor scale longitudinal patient trajectories suggested an approximately linear decline in function over time in most patients. It was observed that higher cognitive or motor scale scores at enrollment were associated with longer subsequent survival in patients with sporadic Creutzfeldt Jakob disease. To quantitatively differentiate phenotypes between prion disease subtypes, the paired cognitive and motor scale score, called a CM ratio, was used. A CM ratio of greater than 1 indicates greater relative motor dysfunction, and less than 1 indicates greater relative cognitive dysfunction. A comparison was done for patients according to prion disease subtypes, and across codon 129 genotype in patients with sporadic Creutzfeldt Jakob disease. The CM ratio for patients with iatrogenic Creutzfeldt Jakob disease was significantly greater than the CM ratio for sporadic Creutzfeldt Jakob disease. Patients with inherited prion disease, caused by the 6-octapeptide repeat mutation, exhibited low CM ratio. The CM ratio may also be valuable in objectively quantifying the phenotypic progression in inherited prion diseases. The median CM ratio for patients homozygous for methionine at codon 129 was found to be significantly lower than those heterozygous or valine homozygous. This indicates greater relative motor dysfunction in the latter two groups. The composite examination scale score, which is an average of each motor and cognitive scale score, was evaluated for 528 patients with sporadic Creutzfeldt Jakob disease. Examination scale score sporadic Creutzfeldt Jakob disease patient trajectories showed a longitudinal linear decline, and higher scores at presentation were associated with longer subsequent survival. Thus, use of rash methodology for development of motor scale and cognitive scale was successful. Both scales are unidimensional, perform independently of age or gender, and have excellent inter-rater reliability. They can be completed in minutes at the bedside, as part of a normal neurocognitive examination. These two scales, along with the composite examination scale, could be useful to objectively measure phenotypic and clinical change in future clinical trials, for patient stratification and for feedback as part of routine clinical care. This concludes our today's Neuro Shorts podcast, brought to you by MedSynapse. Thank you for listening.